Hi guys, today I am again with a new concept, that is recessive epistasis. The concept as epistasis was introduced by William Bateson in 1909. They noticed that certain gene combination did not follow the expected Mendelian ratios that we have already seen in monohybrid and dihybrid cross. Because one gene, that means there will be two genes relation in which there is a one gene could mask or modify the effect of another gene, and this masking interaction was termed as epistasis. So we're going to look forward for a very amazing example, that is our blood group. Before any further explanation, we should know why these two genes are responsible for. H gene, that is located on the H locus, is responsible and encoding for H compound. H compound is actually converted from any intermediate source in our body. So if H gene is not functional in our body, that means it will not encode for H compound. And why we are again and again talking about H compound? So H compound is important for attaching sugar to the terminal of A and B group. For example, if somebody has two alleles A and B both, so H compound is missing, then there will be no attachment of sugars to these alleles. That means if there is no attachment of sugars to that allele, it will not express in our body. That means by default our body will show O group. Understand? Also, ABA locus are responsible to encode sugar for the specific A, B and I alleles. For the A alleles, N-acetyl galactosamine sugar encodes, and for the allele IB, the galactose sugar encodes. Remember, without the help of H compound, the sugar will not attach. So H compound should be there, okay? Now, there are two ways of having O blood group. The first condition is Bombay phenotype, that is homozygous recessive condition. In the H locus consists two H alleles. If there is a 2H allele, there will be no formation of H compound and there will be no attachment of sugar to the A and B allele. If there is a no sugar attachment, there will be no expression of A and B group. Condition will be O group phenotype. The second condition is if we have this II allele, that means compound is present but there is no sugar added. That means if there is a condition we get from our parents, so ABO gene who are responsible to encode for the sugar will not able to encode any more, okay? That means this condition, homozygous recessive allele II, will halt the ABO gene functionality and there will be automatically O group phenotype.